Hey exobiologists, welcome to the crew. I'm Lord Janda, and this is my Let's Play of In Other Waters. We've just finished exploring the West Reef and we've got a microbial power cell that upgrades the engines on our diving suit now. So we're going to go ahead and mop up these four samples that are scattered around the east and east central reef before we head north and advance the story. So we've got damaged or exposed sing stalks, table stalk growths, reef caps, and shimmer blooms. So the first thing, I might not be able to keep track of all these in my head. I mean, I know it's only 4, but sue me. We've got to go north first. This way. I think I know where that is. Okay, so you'll find the controls and the utilities where sampling and everything else is. Once you've found the propulsion system, let's try it out. But remember, it uses oxygen. Be careful. When you've got the hang of it, let's try to cross the northern rift. Prime before activation. Or does it just use oxygen? I really thought for some reason that it needed fuel in the form of samples of stuff. This is what we need to go up, I think, to get to a couple of those samples. Yes, hello little things. I've run into you before. Oh. I think I'm gonna have to hit these with a shrill sack or something. Yeah, that's what I thought. I'm gonna have to get a shrill sack or something to hit these with. Don't really know where the nearest one is either. It's annoying. Is that one? No, that's a sing stalk. That'll not have them. I think we're probably gonna go this way. There were a lot by the forest. This one? No, that's a signal. Ah, wait, here's one. Okay, now east. Sorry, guys. Hate to frighten you all off, but... I can grab another couple of shrill sacks here to replenish the supply. Is it not? Is it this over here? I gotta maybe go further. Yeah. Oh, there we go. Aha. Finally, the stalk scraper. We're naming them that for obvious reasons. Yep. A large creature with a translucent shell and jointed legs for swimming and feeding. Graces on the membranes of reef stalks. That actually could be symbiotic also. Like, maybe they're cleaning type species, you know? Yeah, I think it's there we've got to get. I could be wildly incorrect about that, though. It's hard to exactly correlate the map with what I see here. The big map, I mean. However, this was clearly implied as an area we could only explore once we'd gotten this engine upgrade, so... definitely to be tried out. Yeah, better propulsion, okay. Does this work then? Are we using it? We are. Okay. The central rift is deep and wide, and the current running down it is relentless. Spores and other particles leave greasy streaks on the faceplate of my helmet as they hammer into it. I gotta read fast, because we're running out of... The suit creaks as I lean into the flow, and the unbroken walls ahead suggest nothing worth risking my life for. Screw it, I'm gonna keep going. Aha. This mushroom-shaped cap cowers beside the rift. Its cap broken, segments of it sit in the sand. We could sample one here. Oh, didn't I do that already, though? Oh, no, I guess not. 10% fungal, 30% tissue. Maybe we didn't. I don't know. For some reason, I feel like we got here. We got this sample before. There's no way further north, though. We got... No. Yes. Consumes bits of oxygen to power this, but not too badly. I'm increasingly certain that I went too far east or the wrong way there, but... I'm gonna check which one of the four vanished, and if it's the one I think it might be, then yeah, we did. Yeah, it was the one that was way down here, so that's where the big rift is. You know, that does mean I need to get into the small rift. Oh, but we get into it just via right there, don't we? Yeah. Okay. I think that's the truth of that. Oh yeah, we can analyze this clouded shell now. And we should be able to unload our cap section samples, section of gilled cap tissue from the storing stock. Let's doesn't always update if the other one hasn't updated yet. Okay, lab, samples. Analyze this. Okay, I think that'll finish the reef cap. Yep, theories. Analysis of reef cap tissue showed that the reef caps maintain a bacterial symbiont within a series of internal chambers, which, while unique to Gliese 667 cc, resembles Epulopicium, a large-celled bacteria found in the stomachs of terrestrial surgeonfish. Epulopicium helps surgeonfish break down algae effectively, and the reef caps bacteria seem to do the same, with traces of algae-like growths found within the cap chambers. Where's this algae coming from? However the caps receive it, this algae is allowing the caps to grow bacterial colonies, which then reduce themselves to endospores when the algae is digested. These endospores are then released to the stalks in the local area to renew the strength of their colonies. It's an incredible recycling system for protecting this ecosystem. Oh, that's cool. So they're absorbing the algae, local algae, and turning it into the spores which they release to the other stalks. Oh, yeah, they are a bit of a... They're more prominently rounded and cappy than the other, mushroom-like than the other stalks. Chitin plate sutures, spore-releasing gills, supported reef stalks all around. Fascinating. Then Animalia novi, Arthropelta, jointed shells, the stalk scraper, Nubilis strigalis. Stalk scrapers are large, translucent shelled creatures with a distinctive swimming motion which feed primarily on stalk colonies. Surprisingly nimble despite their size, stalk scrapers use some of their modified limbs to swim through the water, and others to scrape away the chitinous membrane of the stalks. This is tolerated by the stalks to some degree, although they will also deploy shrill sacs to scare the scrapers away. Stalk scrapers seem to be one of the more socially active of the reef creatures, and will call to each other when grazing or when frightened. Each specimen has a unique cloud pattern on its shell. I wonder how these are formed and what their significance is within communities. Oh, I need to analyze the food sample analysis, yeah. I need to grab the... Pretty sure I still have it in here. Okay, this stalk bark that I've been trying to use as fuel for ages, because some half-remembered nonsense. I really just need to analyze it for this purpose, and I think that's probably the end of it. Multiple entries updated? No, I think one of them is the stalk bark, but I don't think that's actually true. Yeah, we've read this one before. However, the stalk scraper, yeah, there we go. Analysis of stalk bark shows it's chitin-based as suspected, containing glucose and high levels of polysaccharides. This makes it an ideal food source for the scrapers, but not only this, it makes stalk membrane a vital substance for lichen scraper shell formation. The more stalk membrane consumed, the larger and thicker a scraper shell will become, which explains the variance in scraper shell size and translucency. Going further, shells may also be markers of a stalk scraper's importance within the community. A larger shell means a more effective grazer, and therefore a more preferable partner. Further analysis of scraper shells is needed to continue this line of inquiry. Well, we got this broken shell. Chemical analysis of a stalk scraper shell confirms that not only are they made from stalk membrane materials, their distinctive cloudy coloration comes from stalks too. Stalk coloration, like most fungal coloration, is reflective of the local water acidity and the medium the fungus grows in. Makes sense. This variance of amber, orange, yellow, and even green pigments is passed directly to scraper shells when consumed, resulting in beautiful whorls and blobs of color. A scraper shell is a living record of every stalk colony it is fed on, and therefore a marker of territory and social standing. Scraper shells could be used to help study the entire reef and its shifting ecosystem, each one a chemical map of the local history, readable for those who can learn the significance of its forms. So they're kind of trilobite looking, huh? Leg extension points, yeah. Cloudy shell markings. Swimming limbs. Scraping limbs. Oh, so they use their front limbs to swim and their back limbs to scrape, sort of, maybe? Maybe it's the other way around. Alright. So then, yeah. To get to these, we need to go just due east from the central reef. And I neglected that somehow. But we got the other one, so that's fine. There'll be 2 samples here and then one way out in the islands off the East Reef, which I'm pretty sure we can access just fine now that we have the, yeah, just across here. Yeah, I was, whatever, we got 1 sample. So we're good. There's one of them on this side. I feel like it might have been. These currents will still block our passage. Not anymore, they won't. Yes, yes. Prime the engines. Let's go. This western shelf is buffeted by currents, bringing with them a scattering of swirling spores. The broken shelf of the reef reaches out towards the deep like a hand, the first of 3 fingers which mark the edge. No, nothing here though? Okay, south then. Here a rift meets the exposed reef edge. What caused this huge slash into the shelf? Surely not just erosion. Nah. Okay. It couldn't, but that's alright. We'll spend more oxygen. Apparently all it costs is oxygen. Remember that properly, I guess. A shattered section of the wall allows passage onto the outcrop. And made that crossing, and I'm pretty sure here is where the other couple of samples will be. This section of the shelf, sheltered by the tall headland to the north, is strangely peaceful. But there's more trouble up there, yeah. Beyond these thickly entwined stalks, a violent current can be heard, rushing down through a gap in the headland. The water's funneled by the rocks into this clearing, making it hard going against the rush. Is that a big... Oh, that's a stock scraper. Okay. A large boulder protects the mouth of this passage from the onrushing waters. Battered by the water, these stalks cling to the rocks. A single creature braves the current to graze on their twisted forms. It sure does. Aha. Attached to the side of the shelf, this canopy is partially bent by the current. If we're careful, we can sample a growth from its top. 20% fungal, 30% seed. The computer's very confused by this, but I would say it is indeed both fungal and seed. A ribbed growth found entwined in stalk canopies. Yeah. Look at this brave little stalk scraper. Now I have to use this to go back to. Doesn't entirely make sense. Should be pretty easy to go with the current. See if there's anything through here. Yeah. Little streams of sand pipe through gaps in the rocks here, driven deep into cracks by the current to the north. This gap between the rocks allows passage both into and out of this exposed second finger of the reef. Another rift separates the second and third fingers of the reef, looked over by a ragged stalk canopy that sheds particles into the flow. We can cross. Current here is both exhilarating and terrifying, like standing on a cliff top and leaning into the wind. But I think it's actually, that would be the wrong way. I think the other thing was at the south end of this second finger. We are burning a lot of our oxygen doing this, but that's alright. hopefully. These huge stones have been worn smooth by thousands of years of sitting beside the rift. White sand is gathered against the boulders on either side of this passage in soft, pale drifts. This tall stalk is torn open, perhaps by a predator. Inside, a rich tapestry of fungal life forms exposed to the open water are slowly dying. Perfect. Nab a question mark, 20% fungal. It's a fungal cluster. Cluster of creatures sampled from the interior of a stalk. Okay, I was just going to go straight on to the east, but we're, whoopsies, wrong button. We've used a ton of our oxygen, so I'll just go back, turn these 2 samples in real quick, and then we'll go to the east and out onto those little islands. So we got a fungal cluster. And we've got canopy growths. And that should wrap up the fungi, actually. For the most part. And we have the sketches for the reef stalks, and the signal stalks, and for the reef bloats, and the reef caps. So now that just leaves the sing stalk and table stalk. Theories on the sing stalk. What did we know about this before? Right, they hum or groan, and they're hollow. That's right, and they're spirally entwined. So we needed to know what was in them. Right. Attempts to analyze the interior of a sing stalk reveals many different organisms within the stalk which are not genetically related to it in any way. It seems the sing stalks are actually tubular gardens which host juvenile fungal creatures. The swaying of the stalks keeps these sealed environments fed with filtered water and microbial life, while allowing their spores to be distributed across the reef. However, I haven't found a single example of any of these life forms growing outside of a sing stalk, suggesting that they can only survive within these carefully controlled tube gardens, and that the spores they release are being used by the stalks for other purposes. Are these vertical farms designed to cultivate other species that might feed the rest of the stalks with showers of nutrient-rich spores? So, well... maybe. Yeah, actually. So they've got sealed environments. The stalks sway and filtered water and microbial life flows in. The fungal creatures inside only grow in them, and then they release spores, which those stalks use. Yeah. These stalks, I'm beginning to think they're pretty damn smart, you know? Like, seems like they might have farms, possibly pets, the ability to intentionally genetically modify themselves or other things. Internal fungal colony. Then the table stalk. Yeah, they're from lots of entwined stalks. They provide a steady flow of water through their canopies. They extract minerals for some purpose from the bedrock that's not for the stalks. Laboratory studies of the growths found in the upper canopy reveals that they contain the genetic material of tens of different species. These growths hold within their round casing seeds from many species local to the table stalks, as well as spores for the reproduction of the stalk colonies themselves. Fed by a carefully maintained flow of the correct minerals and chemicals, these seeds and spores are preserved by the stalks, kept viable for long periods. Could these seed banks be a complex example of collective serotiny? On Earth, some plants hold viable seeds until an environmental trigger such as a wildfire or disease causes their release. Perhaps the stalks are preserving the local ecology in case of environmental disaster? Are they the guardians of this reef? Okay, so they also have seed banks. I mean, not that, yes, Some plants on Earth, for instance, do only release their seeds after wildfires and such. But yeah, these stalks are pretty intelligent, I think. Entwined canopies, 8 to 12 meters in height. Oh, those are big, yeah. Captured seed banks, color and type variation within the entwined stalks. And that's a reef stalk for scale. So yeah, these are proper trees, basically. Okay, so that's all the stalks cataloged. Now, there was one plant which we haven't cataloged properly, fully, and that's going to be what we're going to find on these eastern islands. Okay, so there's the islands. We are pretty much gonna head out eastwards. Let's see if we can find our sample on those islands. It's gonna be for the petals. A strong current cuts across the east edge of shelf with the turquoise haze of the open ocean stretching beyond. Alright, let's power up the old microbial fuel unit. And let's go. East of here, the current fades, and strange pillars rise up from the dark of the ocean floor. This large pillar extends down into the murk of the deep water below, standing improbably straight and tall, a geological anomaly. Also, there's plant matter. These long, pale green strands of plant matter entwine in flat blades which trail horizontally between the pillars. Oopsies. Ran out of fuel. A little bit. We're fine, we can make it. This pillar is topped with a layer of bright, sunlit life. Looking around, each of the pillars here has the same green-gold peak. Okay, so whatever doesn't live down there does live up here. And there's strands of silken plant matter running between them, which we haven't encountered before. These silken strands meet in places and become tangled in one another, forming complex webs that are slow to navigate. Ooh, creature tracks. Patterns of dents in the sand show the paths of the feathery creatures that cross from pillar to pillar in slow and purposeful loops. We literally haven't found them yet, but apparently I found their tracks. 5% chance of being an animal. Well, probably higher than that. A shed feather found in the pillar gardens. Oh, there they are. A cleverly camouflaged creature, mainly composed of feathery, frond-like arms, which waves rhythmically as it moves. Well, aren't those interesting? These funny feathery creatures are all around these gardens. Let's start observing them. Okay. Ah, shimmering petals. In the sunlight, these spade-like petals open themselves wide to the light, welcoming it gratefully. These trailing plants can be of great length and appear to only grow from the sides of the pillars. How did they form? I think we already have petal samples. Yeah, we have pollen. That's not the sample we need. Between the pillars, bright green webs of twisted tendrils extend, forming thin pathways from garden to garden. Fascinating. Observing the underside of these plants, I can see a layer of pores, stomata perhaps, for waste release. The green tendrils that close this space seem to calm the currents, creating a pool of still water. Yes. More shimmer blooms. I've observed these plants in shades which stretch from bright green to ivory white. Is this an indicator of their age or health? The green tendrils don't seem to reach east beyond this point, preferring instead to turn back into the protected waters south of here. Well, let's go beyond. Out away from the pillars, the ocean grants a sense of vertigo, fading away to unknown depths below. And this, I believe, is the last. This pillar, unlike the others, lacks a garden of its own. Have the plants been cleared, or have they simply not reached this far yet? This is the furthest and most barren of them. What's all that? The light of the system's 3 suns falls onto something amber and huge to the east, almost like a pale orange cloud. Oh, that's something, all right. This translucent tentacle doesn't have a fixed shape. Its individual parts instead cycle through developmental phases as they cohere. That's a lot of life right there. What's this then? These golden yellow streams of life shift around solid objects held suspended in the water. Are these bones, rocks, or something else? This creature is impossibly large. I don't even know how to start logging data on it, but I'll try. So this is a single floating tentacle colony creature. This hard barrier is webbed with pulsing nodes. To gain access, we would need to slice through it somehow. Oh, okay, so we can't go further here yet, I don't think. But damn, there's a lot of life here. What are we looking at here? This appears to be a huge colonial life form. There are passages inside of it, but unless we can cut this barrier, we can't reach them. Yeah, there we can't, so we'll just study these quick. This golden colony is overwhelming. Where to even begin? Thousands, maybe hundreds of thousands of zooids make up its shifting interior. It seems that the zooids are split into specialized functions and forms, from jellyfish-like medusoids to balloon-like bladders of gas. So there's a massive colonial life form of some sort out here, but we can't explore it properly because we don't have, we're going to get another tool, I guess, at some point. For now, back into the garden here, we can study these plants that hang between the pillars and the feathery creatures. A couple of bonus life forms there. This pillar top is thick with waving petals, each one caked in tiny particles of golden pollen. This is just another sample of pollen. I don't need that. Nope. I mean, yes, but so I don't need it. Ooh, these entwined tendrils are flat, like blades of kelp, and hold themselves taut between the pillar tops. And I can sample them. Question mark. 5% plant, 5% tissue. Silken strands, pale green strands of plant tendril from the pillar gardens. Okay, this way. Oop, there's another feathery thing. They hang on to the things, it looks like. They travel along those paths. It appears that a number of the creature's feathery arms have sharp edges that it uses to prune the pillar garden's flora. That's interesting. I've noticed these strands don't connect directly to the pillars, but disappear into holes in their sides. What are they connected to, then? Well, that's an interesting question. What dictates which pillars the tendrils stretch to and from? I've yet to see a tendril only halfway formed. There's a sample here, but I assume it's just more, silken strands. Okay, we're calling this sea silk. So are you going to say something? Yes. Named this sea silk, though I'm not sure what kind of plant it is. Check my notes. Yep. Long strands of pale green and white plant matter that twist into broad, flat bridges and webs between the pillar gardens. Yeah, that's pretty unique. Oh, the lower parts of this pillar are marked with narrow holes. Along them, small plates of chitin shells glint. Okay, is this sample going to be for the plants or for the animals? Oh, startled something. Got it. Burrowing creature. When disturbed, this segmented creature shot out of its burrow, ejecting grit and sediment as it swam away. Quite the surprise. Yes, they are tricky to catch a glimpse of. For sure. What can we sample here? Question mark. 20% animal. 0% shell. It's, a chitin plate. A shard of chitin plate left behind by a segmented worm. Interesting. This broad pillar's full of rough-looking bore holes. Chitin plates glinting here and there suggest they're burrows. So do these grow between the burrows of whatever this worm creature is? Is this more? Yeah. Is that another one of the feathery ones? Yeah. I've observed these feathered creatures moving between pillars, along the web-like plants that connect them. Yeah, they do move along the webs. This patch of water is entirely enclosed by webbed tendrils, and thin strands float through the water like lost hairs. I don't think there's anything we haven't seen up here, no. Gaps between the tendrils are webbed with fine strands, almost invisible to the naked eye. Okay. These two tendril bridges almost meet before disappearing into holes in the pillar. Are they rooted to the rock inside? What can we sample here? I thought there was a sample. Oh, no, it's not. It's just a feature. Okay. The gaps between the pillars are a pattern I cannot read, one that speaks of a complex geological history. Yeah, that makes sense. This impossibly thin pillar rises up from the murk like a pencil, its sides marked by the faint strata of geological eons. I haven't seen any of the other burrowing creatures. The feathery creatures that pass along the tendrils seem to be unshakable in their routines, each one carefully doing its rounds. There's one. It seems that if they're knocked off a surface, these creatures can swim, but very inefficiently. It must be an emergency measure. It's apparent that matter from the blooms, such as pollen, collects in this creature's feathery arms. Is this intentional? Maybe, yeah, if they're feeding that way. Some of these pillar tops are very small, but that doesn't seem to stop the petals colonizing them with their bright shapes. Oh. Got it. These creatures scrape away at the pillar surface, carving their burrows into the rock. It's impressive given the density of the pillars. We're calling the feathery creatures gardeners, then. They're attentive and purposeful. A feathery, slow-moving creature that prunes the plants of the pillar gardens, moving between pillars along their silken bridges. How did these monoliths form? Perhaps they're cores of harder rock which remained when the rest was eroded away. Yeah, could be. The individual thin, fibrous strands which make up these pillar bridges are so fine they could almost be silk. It's easy to get confused among the webbed tendrils, navigating between sunken islands of life. Fortunately, I have this giant map, so I'm not actually very confused, to tell you the truth. The tendrils seem to be avoiding this pillar. In fact, they appear to be avoiding the main shelf of the reef altogether. That's true, they aren't connected to the main shelf, and it's not really that far. This tendril has to stretch a long distance to bridge these pillars. How do they grow like this, without any support? Yeah, this one's pretty long. Too small to enter, this hole in the pillar leads deep into the rock. A tailpiece left by the inhabitant sits just inside. We could sample it. Fantastic. Are we about to startle one? Yep. Nearly missed that one. I've observed these creatures have a bright tail lure made from many thin, pointed plates. What's its purpose, I wonder? I don't know, but I imagine we'll find out because I'm about to grab one. 10% animal, 5% tissue. Shed tail. The bright chitinous shards but tail shell. Shed or severed. Where these tendrils meet, the feathery creatures are able to carefully clamber from one to another, leaving behind feathers as they go. Oh, does that mean there will be feathers here? Although, we have feathers, don't we? Yeah. This is shed feather, but that's fine, we have one. I thought we had this. These tendrils have grown close enough to each other to form a complex tangle of strands between the small pillars. Okay, we were up here otherwise before. This pillar top's a little sparser than some of the others. Perhaps these more southern pillars have only been recently seeded. Perhaps. This isn't the core of the colony. It's still just pollen, though. Despite sitting all the way out here, this pillar is hooked up to the others by a long tendril. Yeah, it is a bit... this is a bit distant here. It's more sparse down here. Ah, a creature has left behind one of its delicate arms here. Perhaps in order to escape from a predator? We could take a sample of it. Oh yeah, sorry buddy. We're just gonna grab your whole damn arm. Wait. Oh, that one I've seen already, apparently. I'm gonna have to drop something. Probably just this. I think it's not actually that useful. Oh, but I've poisoned the water. They'll love me for that. Okay. 40% animal, 5% tissue. I'm a very bad scientist, aren't I? I did just drop a random sample of harmful microbes into the ocean. A feather arm clogged with pollen from the pillar gardens. Okay, can we get to the furthest south one? Down here, only a single bridge leads from pillar to pillar. A lone creature braving the currents as it crosses. Out, away from the other gardens, this wide pillar feels like a calming sanctuary. A blob of green and gold in a sea of blue. There are still... Are there more pillars? Or what's this lead to? A tendril loosely dangles from the pillar, its root half pulled out of the rock. It's pale and bulbous. We can sample it here. Oh, there's one more pillar down there, at least. Okay. Okay, I'm gonna have to dump. Let's just dump the other spark colony. We still have more at home. Sorry about the poison, guys. Not my intention, but you know. 20% animal, 20% tissue. Animal! Interesting. This should have been a silken root. A spherical, translucent ball. Is it a root? Should be plant, though, you'd think. The last tendril. One final bridge stretches down to the southernmost pillar, its strands swaying with the rising current. Despite being so thick, this pillar is cut through with small holes, and on its top a couple of shell plates catch the light. Okay, we gotta be ready to nail the thing if it comes out. Yep. There we are. These long pillar worms. They seem to have burrows all over this place. These long, segmented creatures with distinctive bright tails burrow into the hard rock of the pillars to protect themselves. And here... these young, small petals are starting to open up to the three suns of Gliese 667 CC. This place will be even more beautiful one day. And here... Okay, I'm gonna have to dump one of these. Fortunately, the chitin plates are duplicated. Oh, I guess I could drop my shrill sack, but... You have your chitin plate back. Let's grab 10% plant, 10% tissue. I think this is gonna be a seed or a seedling or something. Petal shoot. Ah, a new shoot of a petal plant from the pillar gardens. I think we have everything from here. I literally pretty much can't carry... Oops, wrong button again. Damn it. I think we do have everything from here. We'll see. But obviously that's a whole sort of separate ecosystem we just stumbled into there. Let's go ahead and unload everything we got from our suit. Hopefully I actually got all the necessary samples. Yes, Setophora, the bristle carriers, and Sectiotinia, the segment worms. Actually, first thing, Clavicula, the tendril leaves. Well, that's another thing entirely. Okay, first thing, the shimmer blooms, right? We needed a shoot sample. We have that now. I know that much. Petal shoot. Theories, right? So they do produce pollen, their petals, lots of small stamens. Their pollen is carried by creatures. Analysis of a shimmer bloom shoot has revealed a coiled stalk which each shimmer bloom possesses, allowing them to control their height in the water. This means that if a shimmer bloom grows beyond the optimum depth for photosynthesis, it's able to extend its stalk and rise in the water towards the surface. However, due to the constant sunlight and effectively 667 cc's tidally locked orbit, shimmer blooms also use this ability to retract themselves away from the sun and to protect themselves from cell damage. These beautiful plants are highly attuned to their environment, and I can think of no better species to give Mine's name to. It's a name that deserves to live on in this place. I'm sorry I can't do more. Oh shoot, I completely missed the fact that this was called Sulcult or Mine. Well, we don't know that she's dead yet, Dr. Voss, but alright. Mine, indeed. Recessed stamens. Extensible stalk. I mean, it's basically one giant petal is each plant, plus the stalk. Which is how they were described, of course. But they're very colorful, apparently, and they grow in dense little clusters like this. Right. The other plant we found was the sea silk of the clavicula tendril leaves of Vibuspons sericus. This bright green plant stretches its silken tendrils in long, horizontal swathes between pillars in the east reef. This plant is characterized by its fibrous, fine, and flexible strands that can be as thick as rope and as thin as human hair, appearing almost white in sunlight. How they form their bridges and why the plant's tendrils do not rise to the surface is unclear. I've observed one plant intertwined with strands from other pillars, and when these multiple strands meet, often in areas roughly equidistant from their respective pillars, they seem to form a web. I wonder if strands from individual plants are drawn to one another. Gives me a sample of the strands and the roots. I believe we have both of those. Hold on. Here's the strands. And the roots are here. Okay, we have 4 more, which means we're dead on it, but we got all the samples, yeah. Behavior. Analysis of a strand of sea silk has shown that it is, in fact, from 2 different plants that have somehow conjoined where they met. Oh, interesting. Moreover, there are younger growths formed near to the end of each plant's strand, demonstrating their ability to form new shoots along their length, which must be why I've observed a web-like pattern in some of the tangles. However, new growths require a good deal of energy. The strands are green on one side, a sign of photosynthesis, no doubt, but their underside is distinctly darker and more porous looking. Are the strands able to absorb matter floating up from underneath them? I need to analyze a root structure or similar to find out more. I was surprised to discover that the sea silk does not end in roots. After finding a damaged strand, I have found it emanating from a kind of silken ball that I initially took to be a root. However, in lab analysis, it was revealed to be an entirely separate creature. The sea silk is a product or plant-like limb of this organism. I discovered the root itself to be able to put down but also retract strands of sea silk from within. It's an unusual fusion of creatures. Is this a symbiotic relation or a parasitic one? Regardless, these curious creatures and their growths are the bridge builders of the pillar ecosystem, turning isolated gardens into a connected network and helping them flourish. Weird. So these balls are growing these sea silk out from them to connect to each other or to other areas. Other pillars. They form a sort of web that connect with each other. I wonder what the purpose of all that is. Twined fibers, new shoots, etc. We then did discover 2 new animals here in Amelia Novi. Setophora, the bristle carriers. The gardener, Plumosis topiarius. Gardeners are feathery, slow-moving creatures that live among the pillar garden flora. Their upper parts mainly composed of densely packed, fern-like arms. Underneath is a body resting on a dense profusion of short, root-like legs that anchor the gardener but also enable it to walk, albeit very slowly. As gardeners move, they also rhythmically wave their arms, though it's unclear if this is for balance or as part of their pruning action within the petal gardens they inhabit. Gardeners don't do anything fast. But they do travel between pillars using the silken tendrils that grow between them. More analysis is needed to discern why and what their relationship to these gardens really is. So we need samples of their feathers and their arm, which we did happen to get. Shed feather. And the whole bloody arm. Analysis of a gardener's feather, which I've called a pinule, has revealed the gardeners feed on the same thing they camouflage themselves as, the garden's petals. The gardener feather pinules have a razor-like edge, with which the gardener cuts away thin sections of petal to feed on, without damaging the plant. Dang. That has to be very fine in order to cut off little bits of petal, like... Thinking of either... yeah. It's gotta be very fine control. However, unlike an actual feather, they contain nerves, suggesting the gardener can deliberately control them for precise pruning and for maneuvering in the water if they become dislodged from a tendril bridge. Quite why they risk such journeys, given their ineffective swimming, remains to be seen. Further study may unlock the puzzle of their purpose within the biome. So they eat the petals, but they also travel between the pillars for some reason. Analysis of a gardener's arm filled with pinules has shed some light on the relationship between them and the pillar gardens. The debris coating the sample is actually pollen from various garden species, suggesting that gardeners are an important part of the cross-pollination process. Makes sense. This means their contribution to the ecosystem is not just limited to pruning the blooms, they help propagate them too. Indeed, given their roaming paths from pillar to pillar, they must have some way of communicating and ensuring that no one garden becomes wilted or overgrown. This could be a eusocial hive structure or a territorial signature they leave behind. Either way, they're a well-coordinated team of marine landscape gardeners. Well done, lads. Oh, and they're very plant-looking, yeah. They do disguise themselves as the plants. They catch pollen in their pinules and transport it between plants. They have simple eyes, thin, flexible limbs. Yeah, they're like a moving plant, but apparently they garden. And finally, the Sectiotinia, segment worms, particularly the pillar worm, Tegumentum hortostella. Pillar worms are long, thin, segmented creatures that live deep in the dark tunnels and recesses of the rock pillars found in the East Reef. They're frustratingly difficult to observe properly, due both to their shyness and their surprisingly rapid swimming speed. They can, however, be startled out of their burrows by consistent tapping on the rock. Once glimpsed, it's evident they have striations that taper to a bright, lure-like tail. Their front half is protected with overlapping rigid plates. They also seem to be burrowing or excavating creatures, ejecting dust and sediment periodically from their burrows. Further analysis is required to determine why they live so reclusively within the pillars. So we need plate and tail samples, which, once again, we do have. Chitin plate. And a shed tail. Fantastic. Complete success for that Pillar Gardens expedition then. Analysis of a pillar worm's chitinous plate shows a creature heavily adapted to its burrowing lifestyle. It seems that the chitinous plates found around the organism's head are precisely grooved, allowing the worm to chip away at existing recesses to create tunnels, and funneling the sediment back towards the entrance. These grooves should also help protect the creature's head from abrasions while traversing the burrows. It's logical then that these bugs create the tunnels that other life, especially the silken tendrils and the petal gardens, require. They must be one of the foundations of this unique ecosystem, turning the hard rock pillars into vertical gardens through their obsessive burrowing. So they're making passages for the roots and such, and for the sea silk. Dissection of a pillar worm's tail has shed light on their hunting and feeding behaviors. The The tail itself is made from many panels of bright translucent chitin. Given the tail's brightness and the way it waves in a current, it's reasonable to conclude that the worm is using it as a lure. It could be an effective tool for the worm to attract small grazers to a tunnel's mouth with, before pouncing from the burrow and pulling its prey into the hole with it. However, the tail might serve another function too, as a countermeasure against predators. Was this specimen bitten off or willingly ejected? Either way, the pillar worm remains good at hiding its secrets within those dark burrows. Here's a sketch of the thing, though. It's a segmented worm, sure enough. It's a pretty good swimmer. It's got some rows of fans, we're calling them, I guess, or sort of fins, flippers. It's got feeding mandibles, tunneling limbs. It's not entirely clear what it eats. Maybe the plants, possibly. Alright, so that is the East Reef fully explored out to there. Now there is the big thing out here that we can't get to yet, but that'll come in time. Next time out, we will head north and into the bloom at last. For now, thank you everybody for watching. Remember to like and subscribe if you did enjoy it. And have a nice day. This is Sea Lord Jenda, signing off.